Hello, my name is Ileana Jelescu. I'm a research scientist at the Center for Biomedical Imaging of the EPFL in Switzerland. And today I will be giving you a lecture on going deep into Q-space as part of the diffusion session Micro and Macro of the ISMRM 2020 Educational Weekend. I have no financial interest to disclose. So I will start off this talk about explaining how we can travel in Q space. So first of all, what is it? Why is it interesting to go deep into Q space, and how can we get there? Then I will briefly mention the low to intermediate Q regime, which of course covers DTI as well as higher order cumulants, and where there can be an interesting link established to biophysical modeling. And finally, I will spend most of the talk describing the insights that were recently gained from going deep into Q-space in terms of microstructure modeling or microstructure characterization, namely compartment selectivity, cell size estimates, whether we should or should not account for exchange, and also a model-free approach that is valid at high Q, which is Q-space imaging and DSI. So first of all, let's travel through Q-space and understand what Q-space really is. So this Q vector here is really just the integral over time of the diffusion gradient that is applied. And if this gradient is constant here during this pulse, of course, then it's just the product of the gyromagnetic ratio, the amplitude of the gradient, and the direction, of course. So these are all vectors. And the gradient pulse duration, little delta here. And what people are usually used to is more the B value, which is the overall diffusion weighting imparted by the sequence. And the B value is nothing but Q squared multiplied by the diffusion time T, where T here is roughly the time between the two gradient pulses in the sequence. Now, there is also a very nice analogy between Q space and the displacement field, as is with K-space and image space here, which encodes for the position, of course, when we think about MR imaging. And so if we sample Q-space with enough samples, we can actually take the 3D Fourier transform and reconstruct here this displacement probability function of water molecules inside that voxel, which can be a very interesting quantity to. And so if we continue with this displacement probability function, how it's then related to the signal attenuation that we usually measure. So in an unrestricted homogeneous medium here, this displacement probability is Gaussian. And if we take this integral here, of course, we end up with a signal attenuation, which is mono-exponential, where the full width at half max, of course, of this Gaussian distribution is given by the diffusion coefficient, which we can then estimate if we can estimate this signal attenuation here. Now, of course, in biological tissue, this probability is non-Gaussian. And so this is where, of course, it becomes non-trivial to express the signal attenuation, but it also becomes very interesting in terms of either characterizing this probability or actually being able to express the signal attenuation in a more complicated fashion that mirrors the complexity of the tissue sufficiently well. So one very, very important concept for this lecture, as well as the following lecture, is that the B value is really a big bag for two distinct parameters. So you have this Q vector here, which is really pretty much your gradient amplitude, because you're trying to keep the gradient duration as short as possible to be in the narrow pulse regime, and this diffusion time here, which is really your gradient separation. And so typically what people do without really thinking about it is that they work at a fixed diffusion time. In clinic, this is also usually a fairly long diffusion time, and then they increase the gradient and thereby the Q value to go from a DTI regime to a DKI and to high B or high Q regime. Of course, there are also other ways to approach non-Gaussianity, is to actually keep this B value constant here and instead vary the diffusion time such that you can actually look at trends for time-dependent diffusion and kurtosis, which also give you information about the tissue complexity as well as typical length scales. But this will be covered in the following lecture. So in this lecture, we will fix the diffusion time and just increase the gradient G. So why is it meaningful to go to high Q? What you see here are electron microscopy slides of mouse corpus callosum, and we can see structures such as myelinated axons as well as unmyelinated structures and extracellular space. If we work at low Q, the warping we are imparting here as a function of space is fairly weak, such that spins inside a single axon, for example, will very likely be very little dephased following the diffusion process, and so you will get no signal attenuation from there. So roughly, you're very sensitive to either very fast diffusing things on very long length scales and not so much to small structures. Instead, if we go to high Q, of course, the warping we are imparting is much denser, and here you're starting to have significant gradient of phase across small length scales and even across a single cell. And this is where you're able to start to retrieve, for example, information about very small displacements, such as below the axon diameter, for example. And you're able to efficiently, very significantly suppress fast diffusing components that you may be less interested in. And so going to high Q thereby allows us to achieve compartment selectivity, cell size estimates, or test asymptotic limits of diffusion behavior. So how do we achieve high Q values? Well, like I said, it's all about increasing the gradient strength. And so in vivo, in the DTI regime, you should stop at about B1, which is 1,000 seconds per millimeter square in older units. And ex vivo, because the diffusion is slower, you can push it up to 2. If you want DKI, you should stop at about B2.5 in vivo and B5 ex vivo. And then high Q starts to be around values of maybe B5 or up to 10 in vivo. And the matching value for ex vivo, where essentially you double the B value. And so if you think about clinical scanners, the maximum gradient strength is usually between 40 and 80 millitesla per meter. So you're able to achieve the high B regime, but really difficultly, well, you can stop at B10, but it's getting difficult, especially for short diffusion times. Connectome scanners, of course, much more proficient in terms of gradients, and animal scanners usually have no problem to achieve such high B values. On the ex vivo side, of course, again, animal scanners, and if you really have extreme dedicated experiments, you can use vertical systems or dedicated gradient systems that create a very strong gradient up to 10 Tesla per meter, but over a very small value. So this would be for something in vitro, for example. Now, what do we make nonetheless of the low to intermediate Q values? So of course, in this regime, we can use the cumulant expansion, which is really a Taylor series of the log of the signal in powers of B. And if we stop at the first order approximation, we have DTI. So you can see that in order to approximate this decay with a linear function, we have to stop at a B value of about 1. Beyond that, it doesn't approximate it well, of course. And we get typical metrics such as mean axial radial diffusivity as well as fractional anisotropy. If we push into Q space a little bit further, so maybe up to B2 or 2.5, we can also then quantify the kurtosis, which then relies on this term in B squared, of course, and we can get maps for mean axial and radial kurtosis as well. And so the minimal data for tensor estimation is 1B0 and at least 6 directions on one shell for DTI and at least 15 additional directions on a second shell for DKI tensor. And what's important to know is that this cumulant expansion actually has a finite radius of convergence beyond which it diverges, of course. And this radius here is independent of the number of terms you are adding to this expansion. So even if you account for a third order term here, you would not be able to go beyond this radius of convergence, which is roughly between B2 and B3 in vivo. So this type of analysis is really limited to the intermediate Q gene. But the major advantage of DKI is really the clinical compatibility. It's extremely useful because it's sensitive to tissue complexity and provides complementary information to DTI in a variety of applications that I've listed here. And it's also very practical. So the standard acquisition would be 1B0 and then maybe 30 directions on 2 different shells. This can now be easily achieved within 5 minutes on a clinical scanner using acceleration such as grappa and multiband. And if you're really pressed for time, you can even go for a much reduced acquisition, for example, just 1B0 and then 9 directions on each shell, which already allow you to evaluate mean kurtosis and axial and radial kurtosis under an axially symmetric assumption. So what about biophysical modeling in this intermediate Q range? So there are quite a few models that actually work with B values up to 2 or maybe 2.5. And we can think of this standard model of white matter microstructure here that has 2 compartments. So one is the intraaxonal water, so diffusion in the axon is assumed to be unidirectional along and the perpendicular diffusivity is 0. And then there is the extraaxonal space with 2 diffusivities parallel and perpendicular to the main axon orientation. And this model, of course, can be expressed analytically. So this is the signal equation for this. We have 2 compartments, and these are the parameters to be estimated. But what's interesting in this intermediate Q range is actually that there is a direct relationship between the signal moments or the cumulants and these model parameters. So there are really analytical equations relating here, for example, the signal moments to these biophysical model parameters. So this means several things that I will detail on the next slide. But these kind of models have been developed in the past year. So one good example is WMTI Watson, which is an extension of WMTI with a Watson distribution. And the other one is Lemonade that even pushes to estimating the 6th order moment and not just the 2nd and 4th order. And so the pros and cons of each of these 2 approaches, so either the derivation from the moments and the cumulants or the nonlinear fitting to the signal equations are as follows. If we go for the derivation of the model parameters based on signal moments, this implies linear fitting for the estimation of the diffusion and kurtosis tensors. So this is, of course, a much more stable fit, and it's also faster computationally. And because it relies on intermediate B values, all these protocols are clinically compatible. So then this also makes the biophysical model compatible or applicable to the clinic. On the downside, however, of course, the estimation of diffusion and especially kurtosis tensor is associated with a certain level of bias and uncertainty depending on the choice of the B values and the SNR. And so of course, all of this bias and uncertainty will then spill over into the estimation of the biophysical model parameters using this direct derivation. And also, because we are in this intermediate B range, you may have limited sensitivity to small features. So there are things that you simply cannot model or that is not captured yet at the kurtosis level. On the other hand, regarding nonlinear fitting of the signal equation, well, as any nonlinear fit, of course, you'll be very sensitive to local minima, and this is also computationally much slower. However, regarding the speed or even the robustness, there are now new alternatives using either Bayesian frameworks or machine learning that can alleviate a part of these issues. On the upside, the signal equation remains valid up to high Q. So you may gain sensitivity from using higher B values to estimate your model parameters, and you may also have access to more advanced modeling because you're able to be sensitive to more advanced or smaller features. However, one thing you have to be careful about if you're going to high Q and dealing with signal equations of biophysical models is that most of these multi-compartment models typically assume Gaussian compartments, and this Gaussian approximation of each compartment may also break down at high B values. So this is something to bear in mind, and also something that needs perhaps to be addressed in the model if it's no longer valid. Now moving to insights from deep Q space, I will be covering all these recent applications, and I hope not to take too much of the remaining time, but all of them are really extremely interesting. First of all, what we can achieve by going to very high Q space is a selective suppression of compartments. So for example, we can suppress the intra-axonal diffusivity of brain white matter. And the question that had arisen in the community a few years ago is, of the two solutions of the two-compartment biophysical model of diffusion in white matter, which one is biologically valid? And it was coming down to either the intra-axonal diffusivity is faster than the extra-axonal or the other way around. And so one group answered this question by actually suppressing the extra-axonal signal using a strong planar filter. So there was a planar encoding in the transverse direction to the white matter bundle. And with stronger and stronger planar suppression, you were just left with the signal inside the axons that were as aligned as possible to the bundle. And they showed that actually with increased strength of this planar filter, you had higher and higher diffusivity along this bundle, which meant that the intra-axonal diffusivity was faster than the extra-axonal. So this was a very nice proof and answer to this. The other thing we can look for is a dot compartment. So a dot compartment is a compartment with zero diffusivity in all directions and no exchange. So if we think about it, could represent something like impermeable cell bodies, but there's always been controversy over its existence and whether it was not just an artifact of the Rieschen noise floor. So again, a very creative way to answer this question was to go to high Q, but also go from a single diffusion encoding where you're traveling in Q space along a specific direction here, which is your direction of encoding. And instead, you're doing a whole trajectory by playing dedicated gradient waveforms here, whereby you're imparting diffusion weighting in all possible directions of space at once. So this is called isotropic diffusion encoding, and it allows you then to disentangle sticks or very thin neurites from cell bodies, because essentially the sticks, if you're attenuating the signal in all directions at once, they will be heavily attenuated because they will also be sensitive to their main orientation here, which is where it has high diffusivity, whereas the small spheres will not barely be attenuated at all, even if you do spherical tensor encoding. So you can see with linear tensor encoding, you may increase the B value as much as you want, up to 15. You have very little difference between these two scenarios where you have just neurites or neurites and spherical cell bodies. Whereas with spherical tensor encoding, you start to see a difference here in signal between the two types of configurations. And so Chantal, who gave the earlier lecture, actually showed that this dot compartment, so this remnant signal after a strong spherical tensor encoding, was mainly localized in the cerebellar gray matter, as well as in deep brain structures such as thalamus. So this could actually really help to create reliable models for these different structures in the upcoming years. Finally, another creative way to achieve selective suppression of compartments or how to use it wisely is to validate the stick model for axons in white matter, which has been used for many years but never really validated. And so this starts from the observation that at high enough B values, all the signal is suppressed except from intra-axonal water. And then if the impermeable stick model holds, then the powder average signal should vary as 1 over square root of B, because here you have your individual stick attenuation. This is the cosine squared between the orientation of the stick and the applied gradient. You're integrating this over all possible orientations when taking the powder average, and this is nothing less here than the error function. So this here can be written as 1 over square root of b at high b values. And so we can test, of course, this behavior by going to high enough b values. And this was also done very recently in human white matter on a connectome scanner. So going from B values to from 7 to 11, be careful here when the axis is 1 over square root of B, of course, the direction is inverted. But you can see when we plot the powder average signal as a function of 1 over square root of B, we do see this very nice linear behavior, which means the stick model holds in white matter in this B range. Now, the other thing we can do with high Q values is to try to estimate typical sizes of structures. And one question that has been going on for a few years is the estimation of axon diameters. So there have been efforts in this direction since 2008 or perhaps even earlier, I'm not sure. And the main problem that has been pervading for years were that the estimates stemming from diffusion MRI of axon diameters were up to an order of 10 higher than the histology estimates. And you can see here, for example, it's been shown, of course, in the corpus callosum that going to stronger and stronger gradients, so from 60 millitesla per meter to 300, you're really improving your axon diameter estimation, and you retrieve this low, high, low trend going from the genu to the body and the splenium. But nonetheless, the estimates were very high compared to what we expect in human axons in corpus callosum from histology. And so the main confound, or one of the confounds, was, of course, from orientation, dispersion, and undulation. This has been pointed out, and the solution now is to solely work with powder average to remove any orientational dependence from the analysis. And the other main confound was, of course, working at the weak Q regime, where there is little attenuation inside axons, like I've shown in the earlier slides of this educational, and thereby the extraaxonal water dominated the time dependence of diffusivity instead of the intraaxonal water. So there is now also quite some body of work showing this. And the solution is to go to ever stronger gradients, and this is also now possible thanks to tremendous advances in hardware. However, the latest light at the end of the tunnel for this estimation of axon diameters has been shown very recently to really be going to ultra high Q regime, where you're suppressing the extraaxonal water, but also you're going so far that you start to see observe a deviation from the 1 over square root of B power law. So you're moving from the stick model to the cylinder, and you're effectively able to measure its radius. So then the powder average of your signal now varies as 1 over square root of B with a corrective term, which is exponential minus B dA perpendicular, where this is now your diffusivity perpendicularly inside the axon. And once you're estimating this, you can retrieve the axon diameter or radius from this estimation of the perpendicular diffusivity. And this has been also shown very, very recently, both in rat and human corpus callosum, where going from B values up to from 20 to 120 or from 7 to 30 in human, you could start to see a deviation from this stick model and estimate the axon diameter, which for once was very much in agreement with microscopy estimates of effective axon radii, where we know also that, of course, these larger axons that constitute the tail of this distribution contribute the most. But so the effective axon radius for microscopy and diffusion, for once, were in excellent agreement. So what about gray matter? Of course, the model there should be completely different, but again, going to high Q values also helps us gain insight as to how it should be different. First of all, we observe experimentally a different functional form from that of impermeable sticks, which has been shown to hold in white matter, like I've explained. So here in vivo in the rat brain, we see that the cortex, of course, follows a very different trend as a function of 1 over square root of b compared to white matter, which shows a roughly linear behavior at high b values. And the same has been shown for humans using a connectome scanner. And so then the question is, should we account for a soma compartment in gray matter or for intercompartment water exchange? What should we alter in the model to explain the signal behavior? And so the first way to approach this, of course, is then to account for soma. And so the idea behind the SANDI model was that beyond, of course, B perhaps around 3, the two-compartment model of axons or neurites and extracellular space fails in gray matter, like I've also shown. And so this SANDI model, which is a 3-compartment model, was very recently proposed. Now, it relies on a very strong assumption, which is that of non-exchanging compartments. And thereby, for the protocol, well, you'd need very high B values, so up to 10, but you also need short diffusion times to neglect exchange, so up to 20 milliseconds. And this is, in practice, not feasible on a clinical system. However, it is on animal systems as well as connectome scanners, so this could be tested. And so this is your 3-compartment signal equation where the extracellular attenuation is assumed to be monoexponential with a diffusion coefficient extracellular diffusivity, DEC. The attenuation inside the neurites is also given by the Callahan formula of sticks because we're assuming no exchange. And then the intrasoma compartment or attenuation, is given by the Neumann formula for impermeable spheres. And so all in all, it's possible to get reasonable estimates of soma density and size using this model. So here you have a soma size as well as density. And you can see you have more neurites, of course, in white matter and more soma in gray matter. But again, this model is not feasible, not applicable on data acquired on a clinical scanner, unfortunately. Now, the other angle of approach is instead of accounting for soma, for starters, try to account for exchange, because in the absence of myelin, perhaps there is a substantial exchange between intraneurites and extracellular water. And by exchange, of course, we mean all possible types of exchange in biological tissue across the cell membrane, be it diffusion through the lipid bilayer, permeation through aquaporins, or transport via ion channels and active carriers. And people have tried to measure this exchange time across the cell membrane for a few years now, and the results vary quite substantially from 10 to 30 milliseconds and up to 100 to 150 milliseconds, so the question is still open. But if we again use the powder average to get rid of any orientation dependence, now this kernel here that we are integrating over directions is expressed as 2 compartments, so we are neglecting the soma. But now we can only speak of apparent fractions and diffusivities here, which are F prime and D prime, because each of those in the presence of exchange are of course functions of all the other model parameters. So this makes for a more complicated model to estimate, but this derivation was proposed by Kärger in 1985, and this is something I've used. So this is something that I will be presenting at this year's ISMRM actually. We're looking here at rat gray matter and more specifically rat cortex using a protocol with B values up to 10. And because we're looking at exchange, which is a dynamic process, we also vary the diffusion time from 11 to 45 milliseconds. And the first step here was to assume no exchange, so set an arbitrarily high value for the exchange time and see what happens. And so we see the estimates for the compartment diffusivities are fairly reasonable and also stable as a function of diffusion time, but we can see the apparent intraneurite fraction actually decreases with increasing diffusion time. So this is really a signature of exchange here, where at the longer diffusion times, less and less water really appears as restricted inside a unidimensional stick because it had time to also explore the extracellular space. And so the second step then, of course, is to introduce exchange and compare the exchange time we estimate from the full Kärger model to the exchange time we can estimate from time-dependent kurtosis, for example, if we assume the only source of kurtosis is that between two compartments. So in this case, we see that the exchange time is also fairly stable as a function of diffusion time. It's somewhere between 10 and 20 milliseconds. And more importantly, we see that we're able to stabilize the fraction. So the intraneurite fraction now becomes almost independent of diffusion time once we have accounted for exchange. And if we look at the correlation of the exchange time derived from time-dependent kurtosis to the exchange time from the full Kärger model, we see that there is a systematic, the exchange time from the full Kärger model is systematically lower than the exchange time from the time-dependent kurtosis. We don't know which is the correct one or not, if any, but it's interesting to see that if we go to higher Q values, which this full Kärger model uses as opposed to time-dependent kurtosis, we have a shorter exchange time. And this may be that we have added contributions of both exchange between the extracellular space and the intracellular space, as well as exchange between the neurites and the soma, whereas in the low or intermediate Q regime, which we need for time-dependent kurtosis, we are only sensitive perhaps to exchange between extracellular and intracellular space. And this is work I will be presenting on Wednesday at 13:45 UTC. There will be a Q&A session associated with the recording of the oral presentation. You are most welcome to attend. And finally, I just want to briefly describe a model-free approach that can be applicable at high Q regime, which also has some advantages for now for regions where we are lacking a biophysical model. So as I've mentioned, there is a Fourier transform relationship between the signal sampled across Q space and the displacement probability function. And so if we sample Q space here in a Cartesian fashion, we take the Fourier transform, we retrieve the propagator, and ultimately the orientation distribution function. So of course the main application for this is tractography. This is not something I will cover today. But what I just want to point out is that it's also an approach that can be useful in regions where no model is available. And this DSI has recently been generalized to multi-shell data. So now we don't have to do Cartesian, which also alleviates many limitations. And so I just want to showcase this recent study here where they were going to maximum B values of between 7 and 10 and acquiring 5 to 6 shells, doing the ODF reconstruction. So you can see even in very complex areas such as the thalamus or the brainstem, you can really retrieve interesting features about bundles and orientations inside each voxel. But also very interestingly, if we look here at the probability at different length scales, so going from 0 to 5.2 microns to 7 microns and 15, we can see that we get very, very unique contrast in different brain regions, where, for example, here the thalamus has a very, very low probability of displacement of 7 micron, even lower than white matter, whereas cortex has a very high probability to diffuse this far. So this is something that we can exploit also hopefully in the future to build appropriate models for complex regions for which we currently have none. And finally, summary and perspectives. So what I've been trying to explain is that there are really two distinct parameters that make up the diffusion weighting, Q and T, and high Q allows us to examine short microstructure length scales. And there are several regimes as we increase the Q value, so we go from DTI to DKI and to high Q, and for each of these regimes we have both model-based and model-free approaches that are available. And what we have recently learned from going to high Q, for example, is that axons can be modeled as fast diffusing sticks most of the time, but that it's very, very hard to access their diameter. And we've also learned that still water is meaningful only in cerebellar and deep gray matter. And finally, that gray matter neurites do not behave like white matter axons, so probably we have to account for cell bodies and for intercompartment exchange in any meaningful and relevant gray matter model. And for the future, well, I think we can agree that we can get very interesting and important insights from going to high Q, but unfortunately, high Q only means, of course, ultra strong gradients, which can only be achieved on preclinical systems or on very dedicated clinical systems, such as the connectome scanners. So unless there is really an improvement in gradient performance for widespread clinical systems, we really need to think about how these experiments can change the game also for clinical applications. And I think one of the main challenges for us is now to develop tools that build on the fundamental knowledge that we've acquired from high-Q experiments and actually make use of it in a clinical setting. So this is it for this lecture. There will be a Q&A session on Sunday at 15 hours UTC, and I do hope that you can log on and ask me questions. Thank you.